Here we go with the second set of notes in our genetics unit. This one is going to be on crosses, specifically monohybrid and dihybrid crosses. In this set of notes, I'm going to have you guys do a little bit of practice with making genetic crosses. So you might want to head on out to the main web page and download the document called Punnett Square Practice and print it out if you didn't grab it in class already. So this is going to be your chance to just make sure you kind of know what you're doing and practice a little bit, get some guidance before we start doing some of these problems on our own for real. So first we're going to talk about a monohybrid cross. Hybrid means we're putting things together. Think of like a hybrid car as a cross between an electric car and a gasoline car. And mono means one. So in this case, we're crossing one trait. And a useful way to figure out what our offspring are going to look like is something called the Punnett Square, which is this guy right here. Something you guys are probably familiar with. It's a tool that used to predict the genotype and phenotype of the offspring. And so we're only looking at one trait. In this case, we're only looking at height. So you can see we only have one set of letters. We're only looking at T's. We kind of split those apart and whatnot. So we're going to practice doing a monohybrid cross first. So you can take a look at that cross, that first set of boxes in your cross notes that you have. So in this example, I've given you the genotypes of the parents. Okay, so if you look over here, these are the parents. Using our proper genotype terminology, both parents are heterozygous. In this case, I'm telling you that the phenotype of tall is dominant over short, right? Because we got a capital letter equals tall, so tall, capital T equals tall, little t equals short. Okay, so when we're setting up your Punnett square, first you draw yourself your four square Punnett square like I have here. And basically what you're doing is you're taking the different genotypes that you have. So if this is a diploid cell, right, and it's going to do meiosis, and it's going to split those T's apart, and these are the two options that this individual could produce in, if it's a boy, let's say, in sperm, right? It only has a big T and a little t to work with, so it has a potential of making a sperm with one big T, or the potential of making a sperm with a little t. And the same thing for this individual, right? These are maybe the eggs that this female is going to make, right? These are the gametes, the sperm or the eggs. Those are the two different possibilities. And basically, to solve these, is you just kind of bring this guy over and bring this guy down and see what they equal. In this case, those two would combine together to produce an offspring that is homozygous dominant. That is, it has two big T's. If we look at the next one, we get a heterozygous individual, right? We've got the big T from here, the little t from here, and now we have a possibility of making one individual that is heterozygous. If we continue along, these guys, again, can make a heterozygous, and then here, if we're crossing little t and a little t, well, that's going to equal homozygous recessive, so you have two little t's. So when we talk about this, there's a couple different ways we can talk about it. The first is to look at the genotype ratio, and this is basically just to count up the different options in genotypes. So we have, for homozygous dominant, we have 1 out of 4 of our boxes are filled with a homozygous dominant. That's this guy right here. So a genotype ratio of 25% homozygous dominant. We can count up the number of heterozygous we have, and there's 2 heterozygous, 2 out of 4, 50%, and 1 individual that is homozygous recessive, 25%. So we have a genotype ratio of 1 to 2 to 1. And what this tells us is if these two were to get together to mate, they would have, let's say, for example, a 50% chance of producing offspring that are heterozygous for this particular gene. We could also look at the phenotype, and we can count up the number of talls and shorts and see what that equals. In this case, 3 out of our 4 individuals are tall, right? These are the ones that have a big T, show the dominant allele. This guy's tall, this guy's tall, this guy's tall. 3 out of 4 are 75%. This one does not have the dominant allele. It only has 2 recessives, so it's going to be short. 1 out of 4, 25%. So our phenotype ratio is 3 to 1. And we could say that these two individuals were to get together and have offspring, they'd have a 75% chance of having an offspring that was tall, or a 25% chance of having offspring that are short. Fairly simple, hopefully. Now, if we were going to do a dihybrid cross, we would be looking at two different traits. So dihybrid cross is a cross involving two different traits. So you look over here, we have two different letters, right? We have S's and we have B's because we have two different genes controlling two different traits. In this case, we're looking at a gene for the color of the hair and also the length of the hair. So how the heck do you do these guys? These are a lot more complicated. How do you do them? So the first thing you need to do is you have to determine the genotype of the parents. This sometimes is difficult, sometimes it's not. In this case, I've told you what their genotypes are, right? You can see over here that both of them are heterozygous for both conditions. Now, if I just told you that you had a guinea pig that was heterozygous for short hair and homozygous dominant genes for the hair color, then you might have to kind of convert those words into the genotype letters. Next, and this is the part that can get a little bit confusing, you have to figure out the different combinations of gametes that you can make. What are the 4 different combinations of alleles, or S and B, that you could possibly make? Okay, so if we look over here, you can see you have 4 different possible combinations because we're looking at 2 different genes. So I kind of think of it sort of like the FOIL method in math, right? So this S over here could pair with this B, and that's what you see down here, big S, big B, or it could pair with this B, and that's what you see right here, big S, little b. Or this S could pair with this B, little s, big B, or it could pair with this guy over here, little s, little b, which is what you see right here. Okay, so you get 4 different combinations. Notice that each combination both have an S and a B. Right? That's the way you want it to work. Remember, you're making haploid sperm or eggs. You want them to have half of the genome that you're working with. You want them to have 1 S and 1 B. You don't want to give them 2 S's because then they're going to meet up with another guy or girl that's going to have an S and a B, and they're going to end up with 3 S's and 1 B. And we don't want that, right? We want 2 of each letter. We want the genotypes of the kids to be fairly similar to the genotypes of the parents. 2 S's and 2 B's in this case. All right, let's practice this a little bit. All right, so grab your practice page, and we're going to kind of do this one from scratch. So in this case, all I've given you basically is the parent phenotypes. So we know that one parent is heterozygous for being long fur and black fur, so we kind of know what their letters are. And we know that the other parent is homozygous long and heterozygous for black. Okay, so we have to figure out what the parent's actual genotypes are. We have to convert those into letters. So to do that, first I like to make myself a little key, and I've done that over here on the right. So in this case, I've just arbitrarily used the letters A and B. Again, sometimes you use maybe L's if L is for long fur or whatever, but I like to just use A's and B's because they're easy to tell apart, and the big A looks very different from a little a, and the same thing with a big B as opposed to a little b. You know, unlike maybe capital S and lowercase s look pretty similar, so I like to use A's and B's. So in this case, I'm saying that the dominant condition is long fur, right? You know that because this guy is heterozygous, so you know it has a dominant allele and it's long. So that tells you that that's the dominant condition. So a big A would equal long fur, in which case the small a, the recessive condition, must be short fur. Okay. And you also know the same thing because you're heterozygous black. So you know that black is dominant, and then so we're going to do a big B for black and a little b for white in this case. So based on that, we can sort of use this to interpret what our genotypes are. So we've got a heterozygous long and a heterozygous black. So you know they're both of those guys are going to be heterozygous. So you know we're going to have something that looks like this: big A, big B, little b. Versus the other parent is homozygous long. So we look over here, long is a big A, so we know that's going to be homozygous dominant, so 2 big A's, and then heterozygous for black. You know they're going to be different, but you also know they're black, so it has to be big B, little b. So that's what we end up with for our genotype. Big A, big B, little b, big A, big B, little b. Okay. The next step in this process is to figure out what the 4 different combinations of gametes are. So remember, each gamete is going to get one of the A's and one of the B's. And so what are the different ways we could pair those up? Well, looking at this first parent, we could have the big A could pair it with the big B. That's what you see right here. Or it could pair with the little b. Boom. This little A could pair with the big B right there, or the little a could pair it with the little b. And that's what you see right here. And we do the same thing for the other parent, although this time we're going to get a little bit of duplication. The big A could pair with the big B right there. Big A could pair with little b right there. Big A could pair with this big A could pair with this big B. So you get the same thing as the first one, and boom, same thing. So you got a little bit of duplication. And now all we do is basically the same thing that we did before, is we pair everything up, right? We bring down the 2 different A's that we're getting from the parents. We're getting this A here and this A from this guy, and then this one's contributing this B and this one's contributing this B. And we usually keep them together. So in this case, we're going to get big A, big B, big B. All right, so why don't you guys go ahead and pause the video and actually go ahead and fill in the rest of this Punnett square. All right, now that I'm sure you guys have all filled in the rest of the Punnett square, I want you guys to take a look at what you wrote down and see if you got it all correct. So here's the whole thing totally filled in. So why don't you guys double check and make sure that you actually got it all correct. So then once you have all of your different genotypes of all your different offspring figured out, then it's time to go ahead and figure out what your phenotype ratios are, or what kind of offspring you could expect to get from mating these guinea pigs or whatever they are. So to do this, I just write down all the different 4 options that you can get. You can have short black fur or short white fur, or you can have long black fur and long white fur, and count up how many you have. Right? So the only way that you can get short fur is to have 2 little A's. So find all the ones that have 2 little A's, and then you also want to find the ones that have black fur that have a big B. So basically any box that has 2 little A's and at least 1 big B, that's going to give us short fur with black color. And kind of the same thing, if you want to do short fur white, you're going to be 2 little A's and 2 little B's. So you find all those and count them up. And then long fur is going to be anything that has a long A, or a big A, and a big B, at least one of them. And then the long white fur is going to be anything with a big A but 2 little B's. And we go through and we count them all up, and this is what you should get. You shouldn't have found any that had short fur. Everything had at least 1 big A. And then you look at the other ones versus black and white, and 12 out of the 16 had long black fur, and 4 out of 16 had long white fur. So our phenotype ratios in this case are 3:1. Okay, so with that, we're done learning about our 2 different types of genetic crosses. I know that the dihybrid crosses can be a little bit hairy and they can get a little bit confusing. This is why we're going to practice with them a lot to help you master that process.